Hello students, welcome to our online video tutorial class. So I think you have seen the previous videos, whatever I have made on the chapter that is living world, and I think all of the queries are actually which are present from that chapter. Everything is cleared by seeing those videos. So today I have decided that I'll be starting the next chapter, that is the biological classification. So in this chapter, whatever we have to study about the various kind of kingdoms, okay, Monera, Protista, fungi, plantae and animalia, about their characteristic features, about their various kinds of body structures, as well as we will be discussing about various kinds of physiological systems of those organisms. Even the classification of each and every kingdom also we will be discussing in this chapter. So let's start. First, in this chapter we have to discuss about what is biological classification. We all know that from the first chapter we actually come to know that the classification is the mechanism by which any specific group of organisms will be segregated or differentiated into multiple small groups or categories, and we are placing each and every organism in different group or different category. So that is commonly known as classification. And this classification system, first it was started by the father of taxonomy, that is Carolus Linnaeus, because in the taxonomy only it has been told that arrangement rule of the organisms, and when we have talked about or when we have discussed about the arrangement rule of the organisms, obviously we have seen there classification is one step. So obviously when we will try to classify any organism, obviously we have to categorize that organism either into domains or either into the kingdoms. So in the first chapter we have seen about the three domain concept which was proposed by Carl Woese, and there we have seen that he has differentiated all of the organisms, including animal, plants, microorganisms, into three different kingdoms: archaea, bacteria and eukarya. And after that three domain concept, the classification mechanism has getting little bit advanced. So that advancement of the classification mechanism we will be discussing now. Okay, first we can see here evolutionary history of the biological classification. First we will be discussing about the evolutionary history of the biological classification. First, the concept or the classification system which was evolved after three domain concept, that is two kingdom classification. Here Carolus Linnaeus, he has actually proposed this specific classification mechanism. He has told that all of the organisms present in the world, they can be classified into two different kingdoms, because he has no idea about the presence of microorganisms. He has only got about the information about plants and animals. That's why he has created two kingdoms only, plants and animal, plantae and animalia. Specifically based on the mode of nutrition as well as presence of cell wall or absence of cell wall, based on these two features only he has classified the organisms into two kingdoms. Again I am repeating, based on the mode of nutrition and presence or absence of the cell wall, based on these two features only he has created or he has proposed this two kingdom classification system. Clear? This two kingdom classification system has been modified into three kingdom classification system, where we can see with the plantae and animalia, Protista kingdom has also joined. Protista means such kind of unicellular eukaryotic organisms. That means here we can understand that Ernst Haeckel, who has actually proposed the three kingdom classification system, he has segregated the organisms in three different kingdoms based on the presence or absence of the cell structure or cell number. Because he has told that in case of Protista all the cellular number is only one single cell, but in case of plantae and animalia cell number is more than one because they are multicellular. So based on the number of cells he has categorized the kingdom unicellular. Within that he has kept Protista, and he has made another category, that is multicellular, within which he has kept plantae and animalia. Next, after Ernst Haeckel, another scientists, they have researched and they have proposed the next kingdom classification, that is four kingdom classification system. There with with Protista, plantae and animalia, they have joined Monera also. And this specific four kingdom classification system was proposed by Copeland. Why he has created four different kingdoms? Based on the feature, that is cell type, or based on the cellular organization, he has actually created two categories. One is prokaryotic cell category, another one is eukaryotic cell category. and there he has kept Monera within the prokaryotic cell category, and Protista, Plantae and Animalia he has kept within the eukaryotic cell category. So based on this specific feature, that is the cellular organization or body organization, he has created 4 different kingdoms, or he has proposed 4 different kingdoms: Monera, Protista, Plantae and Animalia. And based on which feature he has proposed this 4 kingdom classification system? Based on cellular organization or body organization. Next, the modern classification mechanism, that is the 5 kingdom classification mechanism, what we are following till now also. This specific classification mechanism or system was proposed by the scientist Robert Harding Whittaker, or R.H. Whittaker. According to him, he has segregated the organisms into 5 different kingdoms based on few features. Whatever the features he has proposed, first of all, cell structure, whether the cell is eukaryotic or prokaryotic. Based on that, he has classified the organisms into 5 different kingdoms. Next, body organization. Body organization means cell number as well as cell type. Both have been considered in this body organization point. So based on that also, he has segregated. Mode of nutrition. Which kind of nutrition is the organism showing? Autotrophic, or the organism is showing heterotrophic mode of nutrition. Based on that also, he has categorized these 5 kingdoms. Next, reproduction. Obviously, which kind of reproduction is shown by these organisms? Asexual or sexual or vegetative. Based on that reproductive mode also, he has segregated these 5 kingdoms. And also phylogenetic classification, that means he has classified the organisms by creating or by determining the relationship of the organisms with their ancestors. So that is commonly known as phylogenetic classification system. So by studying the phylogenetic classification, that means by studying the relationship in between existing organism and extinct organism, he has done, or he has proposed this 5 kingdom classification system. So that's why this 5 kingdom classification is a well modified version of the classification system. So for that reason we are following this. But students, one thing I want to say here, that 5 kingdom classification system is also getting obsolete day by day, because today or nowadays we are following 6 and 7 kingdom classification system. So that has been getting more advanced. The Monera has been segregated into multiple categories again, and that has created actually the 6 kingdom and 7 kingdom classification system. So 5 kingdom classification we are not following, that I can't say we are following, but with the 6 and 7 kingdom classification system also we are following now. Now I am coming for just the differences in between these 5 kingdom organisms or 5 kingdoms. Whatever the different features or whatever the differences are actually present in these 5 kingdoms, let's see. First one, I have taken the point cell type. Based on the cell type, I can say, or you also can see, that Monera, Protista, Fungi, Plantae and Animalia, they have been segregated. Specifically, if you consider Monera, we all know that Monera is containing bacterial cells. So obviously bacterial cells are all containing primitive nucleus. That's why they are all prokaryotic. But starting from Protista and up to Animalia, all specific organisms are containing the 2 nucleus containing cell. Which nucleus is containing nuclear membrane? Nucleolus is present, double membranous cell organelle is present in between those specific organisms. So automatically, starting from Protista to Animalia, all organisms are kept within the category that is eukaryotic cell category. Cell type, you can consider this one as a body organization also, this point. Cellular number, that is based on the number of cells also, Robert Harding Whittaker has segregated the 5 different kingdoms. How? Specifically in case of Monera and Protista, if you will see, maybe the Monera is prokaryotic and Protista is eukaryotic, but they are unicellular, that is single cell containing. But starting from Fungi to Animalia, if you will study, you will see that they are all multicellular organisms. That means the various kinds of organ level or tissue level or systems or organizations it has come. So obviously, based on cell number also, these 5 kingdoms have been segregated. Next one, based on the presence or absence of the cell wall. Specifically, we all know that Monera, that is a bacterial cell, they are containing the cell wall outside of the plasma membrane or cell membrane. And that cell wall is commonly made up of a substance that is commonly known as peptidoglycan. What is peptidoglycan? It is actually the combination of polysaccharide and amino acid. Polysaccharide is a complex carbohydrate or complex sugar, and amino acid. So when the polysaccharide and amino acids are combining with each other, automatically the peptidoglycan structure has created. So in case of Monera or bacterial cell, this cell wall is made up of peptidoglycan. In case of Protista, if you consider, cell wall structure truly it's not present, but for few organisms pellicle named body covering is present. So for them cell wall is absent, but pellicle type or pellicle named body covering is present, that is hard body covering. Next, fungi. In case of fungi, if you consider, obviously they contain cell wall, and that is commonly made up of chitin. Chitin is such kind of complex carbohydrate or polysaccharide. Next, if you consider plantae, that is also containing the cell wall, and that cell wall is actually made up of cellulose, and cellulose is also a complex sugar made up of multiple glucose units. And the last one, if you consider Animalia, obviously they don't have the cell wall because they have only cell membrane or plasma membrane as a covering membrane. Extra cell wall is not needed by them. If you now come to the next point, that is the nuclear membrane. Already in the first point we have discussed that which specific cells are prokaryotic, obviously nuclear membrane will be absent for them, but which organisms or which specific type of kingdoms are containing, or they are eukaryotic organisms, obviously they should contain the nuclear membrane. So as Monera, their nuclear membrane is absent, that's why their nucleus is scattered within the cytoplasm. That's why that is commonly known as nucleoid. But starting from Protista to Animalia, all organisms have nuclear membrane because they have well developed, well structured nucleus with the various other complex structures present in the cell. Last point here we can see, that is a mode of nutrition. If you study about the mode of nutrition starting from Monera to Animalia, you will see that in case of Monera, they are showing autotrophic mode of nutrition. Why? Because few of the bacterial cells are containing chlorophyll, which are commonly known as bacteriochlorophyll. So due to presence of that bacteriochlorophyll, bacteria can obviously perform the photosynthesis, so they can be autotrophic like the plants, as well as most of the bacterial cells, except these autotrophic bacterial cells, most of the bacterial cells, they are heterotrophic because they are absorbing nourishment or nutrients from the host body. So obviously they can be heterotrophic, and within heterotrophic specifically they are parasitic. Okay, because they are actually acting as a parasites, they are causing diseases. Next, if you go for the Protista, the mode of nutrition you can see here in case of them also autotrophic and heterotrophic. Why? Because Protistan organisms are also sometimes containing chlorophyll. Okay, like the Euglena, they are containing chlorophyll, so they can perform photosynthesis, as well as they can be also heterotrophic also, because they are also showing the type of nutrition where they are not making the food, they are dependent on the others for taking the food. So obviously they can be heterotrophic also. And in case of Protista also, heterotrophic mode of nutrition means parasitic. Okay, parasitic mode of nutrition they are showing. Come to the fungi. In case of fungi specifically or mostly or for every fungi you can see the mode of nutrition is saprophytic. Why? Because they can't perform the autotrophic mode of nutrition because they don't have the chlorophyll pigment. So under heterotrophic also they are actually showing the saprophytic mode of nutrition. Why? Because they are mostly dependent on dead animal and plant bodies for obtaining the nourishment or nutrients. So obviously they can be considered as a saprophytic mode of nutrition showing organism. If you go for the plants or plantae, mostly all of the plants, they are showing autotrophic mode of nutrition because they have the chlorophyll pigment, and for that reason they can perform photosynthesis. But few plants are there, as an example insectivorous plants. For them obviously you can write here the plants can be also heterotrophic because they are just absorbing the essential nutrients from those insects body, so automatically they can be heterotrophic. And the next, Animalia. In case of Animalia, mostly they are heterotrophic. They can be saprophytic, they can be parasitic, they can be symbiotic, they can be holozoic. So any kind of mode of heterotrophic nutrition they are shown. They are actually showing this specific Animalia. Okay, so I think the mode of nutrition for each and every kingdom is very much clear to each and every one. Next we will be discussing about the special features of kingdom Monera. Now, that means now we are entering within the kingdom Monera. In this video we are entering within the kingdom Monera, I am saying, but in the next video we will be seeing about the classification of kingdom Monera entirely. So just I am starting the kingdom Monera. Basic features of all kingdoms we have studied. Now some special features only for kingdom Monera we have to study. First special feature is reproduction. That means whatever the type of reproductions are shown by kingdom Monera, that means bacterial cells. Bacterial cells can perform asexual mode of reproduction. Obviously that means they can perform the process that is binary fission. That means a single mother cell is getting divided into two daughter cells, and like that they are reproducing generation after generation. Okay, so asexually they can reproduce as well as they can show the sexual mode of reproduction. How? They can perform conjugation, transformation and transduction. What are these things? These things are actually conjugation, transformation and transduction. These three are under the sexual mode of reproduction, where main target of the bacterial cell is to transfer the genetic material from one specific individual to the next generation individual. So how they are doing that? In case of conjugation, if you think two bacterial cells are coming very close and they are adhering with each other, one specific bacterial cell has the plasmid DNA, that is the genetic material. That plasmid DNA will be transferred to the another bacterial cell, which specific bacterial cell doesn't contain it. So conjugation is the mechanism where both of the bacterial cells are adhering with each other, and by that, by making a tubular structure, that is commonly known as conjugation tube, from one bacterial cell the plasmid DNA or the genetic material is getting transferred to the another bacteria which doesn't have that. Next, if you consider that transformation is the mechanism where one specific bacterial cell is again providing the plasmid DNA to the another bacterial cell which doesn't have this kind of genetic material through the culture media. Suppose you have prepared a culture media of the bacterial cell where few bacterial cells are containing plasmid DNA, few bacterial cells doesn't contain. So on that time, which specific bacterial cells are containing plasmid DNA, they are commonly known as F plus bacteria, fertility plus. Why fertility plus? Because they have that plasmid DNA. For that reason they are fertile. But which bacterial cell doesn't contain, they are infertile. That means they are commonly known as F minus. So from the F plus bacteria to the F minus bacteria, the plasmid DNA will be transferred. Through which? Through culture media, where you are just cultivating those bacterial cells, or you are just rearing those bacterial cells. Next, transduction. Transduction is another mode of sexual reproduction where from one bacterial cell, specifically from the F plus bacterial cell to the F minus bacterial cell, the plasmid DNA will be transferred, but that will be transferred by a specific type of organism, that is bacteriophage. So bacteriophage is taking the charge here to carry the genetic material from the F plus to the F minus. So in these three kind of reproductive modes, we have seen one thing: the transfer of the genetic material is actually the target. So when the target is transfer of the genetic material, obviously this kind of reproductions commonly we can say obviously as a sexual mode of reproduction. Next thing is that if you consider the excretion in case of bacterial cell, you will see that in case of bacterial cell, they are storing the excretory materials or waste products within some specific vacuole-like or vesicle-like structure. Those are commonly known as inclusion bodies, and within those inclusion bodies, when they are keeping those excretory materials as a stored matter, according to requirement, those inclusion bodies are fusing with the plasma membrane, and those excretory materials or waste materials will be just diffused out by the process of exocytosis. Exocytosis is the process through which those specific materials will be diffused out. Third, respiration. Mostly they are containing cell membrane, and after that cell wall is present as a protecting membrane. So cell membrane or plasma membrane is actually performing as a respiratory structure, and that specific plasma membrane through which the respiration is happening, that is always happening through the diffusion process. Okay, always the exchange of the gases in between environment and the organism is happening. And the last point of the spatial feature, that is the locomotion. In the locomotion point, you can see they are mostly locomoting by the help of locomotory organs like cilia, flagella. Sometimes they are using pili and fimbriae also. Pili and fimbriae also they are using. Here I am writing pili and fimbriae. These specific structures are specifically responsible or required for reproduction, specifically for the sexual reproduction during conjugation mainly. But after that also this pili and fimbriae structure is also helping the bacterial cell for the locomotion with cilia and flagella. Cilia and flagella is the main locomotory organ, but that these two are also helping as an accessory locomotory organ. So I think this is the introductory part of the chapter, that is the classification systems. So with that, the 5 different kingdoms, whatever their differences in their basic features as well as the special feature of the kingdom Monera, I think you have understood very nicely. I think you have enjoyed this video a lot also. Thank you.